Thank you for joining us today. I'm delighted to be able to share with you the RSV-B model data that we've been actually developing here at hVIVO. So I take you through how we actually isolated the virus, how we produced a GMP manufactured version of the virus, and then also show you the clinical data associated with that, really setting up the model for vaccine and antiviral testing. And this is the world's first RSV-B challenge model. So we're really excited to be able to share this with you today. At hVIVO, we're really well known for developing challenge models and doing a lot of extensive challenge model work over a couple of decades, actually. But actually, to do those models, it takes a really big recruitment effort. So to recruit a volunteer for a challenge study, what we're doing, the very first thing we do is to look at their blood levels and ask the questions, what is their pre-existing immunity against the virus? Because the viruses that we use are naturally occurring wild-type viruses. This means that all of us have been exposed to some extent to these viruses or viruses very similar to them. So we're all going to have some level of pre-existing immunity. And of course, in a challenge study, we need to be able to see infections to be able to tell whether the products we're testing are going to be efficacious or not. So the very first thing we do is to take that antibody levels and work out what that is. That requires a very huge recruitment expertise and bandwidth to be able to do that. So having the recruitment services to be able to deliver against challenge studies actually positions us really well to have a recruitment ability to be able to offer recruitment of site services for other types of trials. So indeed, that's what we'll also do, is we offer those site services, we offer that expertise of that recruitment team to be able to recruit for your trials as well. In addition to doing challenge studies, we also do site services of phase 1 all the way through to site services on larger stage studies, so phase 2 and phase 3, for example. Now, right from the very beginnings of hVIVO, hVIVO has always had its own dedicated laboratory. In fact, it started off as a laboratory-based business. So we have a very specialist virology and immunological laboratory. And in this laboratory, not only does it analyse all of the samples coming out of hVIVO's challenge studies, but actually it also analyses samples for many other people's field trials as well. So really a specialist on-site laboratory able to work very well for these clinical trials. Now, at hVIVO, we have a really good background of doing challenge studies over the past couple of decades. We've done over 80 challenge studies to date. So this is really world-leading performance. Now, about half of those have been in influenza, about half have been in RSV, but we've also done hMPV, SARS-CoV-2 and malaria as well. And now we're also developing bacterial challenge models, which we'll be able to talk to you about at a later date. Now, this new RSV-B model that we've got, we're really excited about this. And of course, to develop a new model, it has to build on the back of something else. So not only have we got good experience in doing flu and RSV, but really does build on the success of the RSV-A challenge model that was so successfully used in the vaccine testing and helping bring in those vaccines to market. Each of these studies shown on here actually got fast-track status or breakthrough status granted by the regulators on the back of the challenge study data. So not only did they get really early key proof of efficacy data from the challenge study, but actually had significant regulatory benefits to be able to fast track and move forward through the next stage later trials much quicker than they may have been able to otherwise. Of course, we're really proud of our involvement at hVIVO in the development of these vaccines and bringing them to market. And of course, the RSV vaccines are now out there saving many lives. So what we've done now is develop not just an RSV-A model, but an RSV-B model, because once these RSV-A vaccines were out there and actually now starting to save lives, what we've seen as a trend in the research community is to go from these monovalent vaccines to now what's being developed are bi- or trivalent vaccines, so either having RSV-A and RSV-B, but also combined with hMPV, of course, a member from the same family, and now also perhaps also with PIV or para-influenza virus as well. So we're seeing this trend to having the development of these combination vaccines. So at hVIVO, we're now very keen to be able to develop a whole testing platform to be able to look at the efficacy for each of those components of these novel vaccines. We already have the RSV-A model. I'm talking to you today about the brand new developed RSV-B model, and now also we have hMPV as well. So the first stage of this is to collect clinical isolates. The next stage is we produce a seed virus, then we move on to GMP manufacture of these challenge viruses, then a pilot test to see if in the clinical trials are we getting the infections we want, and then we move on to make sure we complete the characterization of the virus to have enough data to power future studies. We've done that now for the RSV-A, RSV-B and hMPV, and we also have an brand new RSV-A that's also been developed. So in this case, it brings us right up to date with another contemporaneous strain. So whilst the RSV-A Memphis has been really successful as a virus, it is also a couple of decades old, so it's not truly representative of what's currently circulating. So we brought that brand up to date with an RSV-A contemporaneous strain as well. And we have these clinical isolates in the freezer ready for the PIV development. So when we start off developing a new model, the first thing we do is to collect patient isolates. So we do what we call our friends and family approach at hVIVO, where we give our staff swab kits with consent forms. They take it home, and then if they have their friends or their family or their housemates have some sort of respiratory disease, they take a swab, fill out the consent form, comes back into hVIVO, and we're able to then diagnose what disease they have. So we do a biofire panel and test against 21 pathogens, and they get a certificate saying you have this little bug, maybe hMPV, maybe PIV3, maybe influenza, who knows. And then we also then can collect those isolates and able then to use them to develop brand new challenge models to meet customer demand. And that's exactly what we did for our RSV-B. In addition to those samples, we also complement those samples in the case of RSV with patient isolates, in this case from pediatrics, who also had those infections naturally in the community. And actually, the sample which got chosen and went all the way through to GMP manufacture was one of those such samples that came from a baby hospitalized with a disease. So you can see this is still very much an important disease that needs tackling. Hence, we're setting up this challenge model to be able to get better and more improved vaccines and antivirals against this really important pathogen. So once we've got all these patient samples from different sources, we then triage them. So the first steps of those triages, does it grow on the GMP cell line? It's very important it's able to grow on the cell line that we're able to use. So those GMP cell lines have to be used instead of R&D cell lines, because we have to make sure we've got no adventitious agents present. Of course, we only want to inoculate people with a pathogen of choice and eliminate any potential for adventitious agents. So we whittle that down to just a few handful seed viruses, and then one is selected for GMP manufacture. Each of those stages all the way through has multiple testing to make sure each of these viruses have all the safety testing. We're eliminating any potential risk from that aspect. So once we have the selected seed virus, it goes into GMP manufacture. And important to note is once we collect the first isolates, they're already aliquoted, so we aliquot them at that stage. This is important because it means when we're developing the methods to grow these viruses, we're not actually going through multiple rounds of production. So we're able to go right back to the beginning. So once we develop our method, we go back to an unused aliquot of the sample and then grow the GMP stock from that. That means we're able to produce a GMP stock with minimal passage. And this is really important to make sure the virus we end up with in the GMP stock has had minimal passage and therefore very little chance to actually adapt to human culture. So it means we're actually giving a virus that's very representative of what was originally circulating in the population. So after minimal passage, in this case 4 passages in the cells, in GMP cells, we're able to then isolate the virus and centrifuge that down to remove the cell debris and then aliquot it. And also really important part of this process is that we, at that stage, even though we don't know the titer at that stage, we aliquot the virus in various different dilutions. And the point of this is it means that we have ready at source different dilutions in very small volumes, so that when it comes to doing the clinical trial, we're able to inoculate a volunteer from a single vial, and those vials have therefore not needing to undergo any further manipulation. So it's GMP right from the source, right to inoculation of the volunteer. Sometimes people do challenge studies where they make very high concentrated stocks and have to dilute at the bedside. This, of course, is error-prone. Each dilution could cause a difference and therefore gives variability in the data. At hVIVO, we make sure that all of our aliquots are directly from the GMP at source with no further manipulations to eliminate that variable. So once we have the GMP stock, of course, it's then vitally important to make sure that the safety of this stock is absolutely paramount. So therefore, we undergo a lot of testing of the stock. So, of course, we do the standard testing like bioburden and the cell-based assays, looking for viral contaminants. But we also do a very large panel of targeted PCRs. So this is a risk-based approach. So we look at the isolated source and think what other potential pathogens could feasibly be in there. So it's a respiratory samples. Therefore, this panel contains a whole list of other respiratory pathogens, human pathogens, that could potentially have contaminated the source, as well as some more longevity type viruses, which also could have contaminated the source, for example, HIV. So we test against a very wide range of targeted panel In addition to that, we also have what we call non-biased testing, so where we're using NGS. So the NGS gives us two things. It tells us about the variants of that virus, but we also use NGS to look for any potential viral contaminant in those samples. So a really thorough way of looking for potential adventitious agents to make sure this is a really safe product to use and only has the pathogen that we intend it to be in present. So then it comes to doing the clinical trial. And these challenge studies in a standard format, so it has outpatient screening, so they come into our screening centres, and the very first question we're asking, what is their pre-existing immunity? Because of course these are naturally occurring pathogens, therefore most of us have been exposed to some extent to these viruses. And in a challenge study, we need the majority of people to get infected, of course. So what we're doing is looking what are their antibody levels like at that point in time. And that really is the very largest exclusion factor from these studies. So once we've done that and ascertained that their antibodies are low enough that they're suitable for the challenge study, then they can move forward, and then they come into our trials and come into what we call a day -2. So if inoculation day is day 0, then they're coming into our study 2 days prior to that. And at this point in time, we recheck their health status. Of course, only the healthiest and most fit subjects go into these studies, because these studies are made safe not by the pathogen choice, but by the selection of the volunteer. These pathogens are actually wild-type pathogens. So if they did get inoculated into a vulnerable person, then they would still cause very severe disease. Of course, that's not intended. In the challenge studies, safety is absolutely paramount. So therefore, the safety of these studies is by careful selection of the volunteers. Firstly, on their immune system, and then making sure they are healthy. They would be expected to clear this virus without any other intervention. So once we've established that, and they're rechecked when they come into the quarantine, there's a period of acclimatization. So they need, of course, be happy they're going to be able to stay in a quarantine conditions for up to 2 weeks. This means they're going to be in an individual isolated room with en-suites, not able to leave that room in that time frame. And our medical staff also need to make sure that they think they're going to be happy, that person's going to be happy as well. So this is a two-way factor to make sure the participant is going to be suitable for the trial. Once that's established, they're inoculated, and then we're able to follow the whole disease time course as it progresses. So we'll start to see symptoms, we'll collect samples for viral loads, and all the way through the time course until at the end of the study, they will then have very limited or if no symptoms at all, and then at that point, they're able to be discharged from the quarantine. And then come back at day 28 for a follow-up visit. So let's look at a bit more detail now about how we actually assess those symptoms of disease. So to start with, a really critical aspect is the symptom diary card. So on this, we have all the potential symptoms we may expect to see for this virus. This is a categorical diary card. So we're asking the volunteers 3 times a day to score their own symptoms. So if they have no symptoms, so no sneezing, for example, they tick the 0 box. If there's just a little bit of sneezing, they may tick the 1 or 2 or grade 3, grade 3 being the highest. Now grade 3, to have a grade 3 symptom, it has to be really bothersome and stopping them from doing their daily activities. So grade 3 symptoms are very rare. Majority of symptoms we expect to see would be grade 1 and then some grade 2 symptoms as well. So in this way, we can not only see what symptoms they're actually having, but also have a categorical and a score against that symptom, so we can look at severity as well. And of course, it's not just from the patient perception, we're also looking at then physicians' assessments as well. So our doctors will be going in there and assessing the volunteers as well. Now, a key facet of respiratory viruses, of course, it does make you have a runny nose. So a crude but very effective measure actually is to look at the total mucus weight. So in this, what we do is we give our volunteers pre-weighed tissues, and when they actually need to blow their nose, they need to use the tissues that we've pre-weighed and given to them. They blow their nose, they put them in a sealed bag, then once a day they're collected, and then we actually weigh the total contents of that minus the pre-weighed tissues, and it gives us a crude but effective measure of how much nasal discharge that volunteer had caused by the virus. In addition to the normal symptom diary card where we have the categorical scoring, we've also been now setting up what we call a patient perception questionnaire. This is a very simple question asking them, how do they perceive their cold is today, and is it better or worse than the day before? This is done just once daily, and I'll come back onto this a little bit more later, as it has quite good value actually, particularly in antiviral testing. So in terms of the study demographics, you can see here the spread of the population that we had participating into this study. Now the The lowest age group, what we had here, the youngest person was 19, the oldest person was 46, with a median of 29. So a very classic sort of distribution for a challenge study. They tend to be the younger demographic, of course, they need to be healthy and not in an at-risk group. And smoking history is a really important facet of a respiratory virus challenge studies, because smoking, of course, can cause risks with respiratory viruses. So we need to understand from a safety perspective what their smoking history is. So the majority of volunteers on this study had not smoked before, but some had smoked a little bit. And of course, with the massive advent of vaping is also a very critical component of that. It needs to be questioned and asked separately. We did have some current vapers at low levels, but majority again had not vaped before. So very clean smoking history, able to recruit those volunteers, making sure safety is their first concern. And indeed, the excellent safety profile that we saw was really, really very good, actually. Very minimal AEs, absolutely no SAEs, and those AEs were extremely minor. So exactly what you'd expect and want to see in a safe human viral challenge model. Now, if we look furthermore now at the data, what we really like to do is look at the incidence of different levels of disease. What we saw with this new RSV-B virus is a really high incidence of disease, actually. So firstly, by PCR infection, if we say how many people met the infected definition, which is looking at the PCR infectivity, and they must have multiple PCRs which are positive within a short time period, actually we saw really high levels of 90% of volunteers met that definition. And then as we go through the disease categories, of course, we're asking for then higher levels of disease severity, which, as you'd expect, you then see lower incidence of that. We all know with respiratory virus infections, you can get asymptomatic infections. So therefore, if you look then at the next level and say how many people had symptoms, we see a slight drop. And then if you look at the lowest level there, actually, we're asking the question, how many had moderate to severe symptomatic infection? Now, moderate to severe symptomatic infection is when we're saying they must not only have the PCR positivity, but actually have recorded grade 2 symptoms or higher on their symptom diary card. So these are really the quite bothersome symptoms, and they would be really very noticeable to the volunteer. Now, this is proven in our RSV-A model and other models like influenza, for example, to be a really good translation to field trials. So if we think about how a field trial is recruited, field trials, people are given vaccines, then asked to go and live their daily lives. And only when they have symptoms that are a little bit bothersome do they then pick up the phone to the researchers or go into their doctors and then get recruited to the next stage of that clinical trial. So in challenge studies, we're very sensitively able to detect the levels of disease, of course, because they're in a quarantine and they're being asked 3 times a day to fill out symptom diary cards. We found actually these grade 2 symptoms, where they're more bothersome, translate very nicely to the field trials, and therefore can give reassurance that is the same sort of population that you would have recruited on a field trial. So this moderate to severe infection is often the endpoint that's used as the primary endpoint in vaccine challenge studies. And we can see here a really robust level of moderate to severe infection that's really reassuring. And if we look on the right-hand side, we can actually then also look at the disease profiles over time. So you can see in the blue, we've got the PCR curve, and in the green is actually the symptoms over time. So as you'd expect, starting off relatively low and then peaking around day 4, day 5, and then coming down towards the end of the quarantine period at day 12. And we've got a good correlation actually of symptoms and PCR. In this case, that the mean symptoms was very slightly later than the mean PCR. And we can see some individuals where that varies, which I'll also show a little bit later. So when we develop a new model, a really key question is, okay, we've got this data on this new model, but how do we know, that's what good looks like? Well, in this case, we're fortunate enough that we have this very well-established RSV-hMPV-A challenge model that has been really successful in correctly categorizing vaccines to those that's going to be successful in later stage field trials. So on the right-hand side, we've put the RSV-A, very successful data, and on the left is the RSV-B. And we can see in every incidence, the incidence levels of disease we can see for the RSV-B virus are always higher than what we've had with the very successful model of the RSV-A. This gives us a lot of reassurance that the RSV-B model is also going to be really successful and really good for vaccine testing as well as antiviral testing. So this is also now shown not just in an incidence, but if we look at the disease severity. So on the right-hand side, we've got the RSV-A model, again, which has been really very successful. And of course, the error bars on this right-hand side are very low, because what we're showing here is a combination of all of the placebo subjects that we have in our database. So a very large N number, hence the very low error bars. And on the left-hand side, we're showing our brand new RSV-B model disease severity. Of course, much lower N number, so slightly wider error bars, but again, very respectable error bars. So we can see actually the disease severity shown by the magnitude of both PCR and symptoms is somewhat higher with the RSV-B model. So by quite a magnitude actually. So this shows us that this model is going to be very good at getting quite moderately severe levels of disease. So really, really robust levels of disease here. So if we look a little bit more detail about what sort of symptoms we're seeing with this RSV-B model, what we're showing on this slide is categorizing it. So we mentioned about the symptom diary card where they categorize 0, 1, 2 or 3, and this is plotted out here. So we're asking, we look back at that data and say, each volunteer, what was their maximum score they scored for each of those symptoms. We can see in the purple are the grade 3, in the yellow are the grade 2, and in the green are the grade 1. So we would expect typically a lot of grade greens, or a lot of grade 1s, but we're actually seeing a really large number of grade 2s. Now this is quite unprecedented. We're seeing a really good, robust grade 2 level disease in the majority of subjects for this RSV-B virus. We're also seeing some grade 3s actually with this virus too. So just as you'd expect, it is predominating for quite a lot of upper respiratory tract symptoms like runny nose, for example, but also seeing really good engagement from systemic symptoms, for example, malaise, tiredness and headaches. And actually there's some level of low levels of lower respiratory tract involvement, maybe cough, for example, but much, much lower levels. And this is exactly what you would expect in this healthy adult human viral respiratory disease, but albeit this is actually very robust symptoms and more robust than many other challenge models that have been developed. If we look now, just a few examples of individual subject data. So each of these curves here, it represents an individual subject other than like what we showed previously, which is a whole group mean. So looking at this, we can see that the symptom profiles vary slightly between the subjects. So in the red bar, we've got the PCR. In the other bar, in the purple, is the infectious units, that's PFU per mil. And then the actual bars are the symptoms that are represented. So we can see each of them have very robust disease, but the one on the left-hand side, for example, the symptoms come up a little bit earlier than the one on the right at the top, where the symptom profile really matches very well to the PCR. And then on the bottom left, we can see the bars, actually, the symptom profiles are slightly later than the PCR. So there's a little bit of variation with symptoms, and this is exactly what we expect to see in the respiratory viral models. Of course, everyone has a different level of pre-existing immunity, and that tends to drive their symptom scores. And of course, and we all know that you can get asymptomatic individuals which have viral load and no symptoms at all. So unsurprisingly, a little bit more variation in the symptoms than in the PCR, but really robust, interesting data. And now, I mentioned right at the beginning there, we also have this other question there about the perceived cold. So in addition to the symptom diary card, which is categoric, they're also filling out a daily questionnaire of do they perceive they have a cold or not. And if we see here, in the blue, it's those that perceive they had the cold, and in the green, those that perceive they didn't. Now, of course, as you would expect, very much higher levels of disease in those that had felt that they had a cold, just as you would expect, but of course it's not zero. So some of them still had very low levels of PCR who didn't perceive they had the cold, and that's because those symptoms really aren't bothersome, so they're not able to detect that they've got that virus, and it's not therefore really bothersome enough that they're perceiving that they've got a cold. Now this comes into its own when you think about how you can use challenge models. We've spoken a lot today about vaccine testing, which is really depicted by A on this graph about giving a vaccine, or indeed giving an antiviral prior to inoculation with the virus. But actually, these challenge models can be used to test products right throughout the entire time course. So in B, that's a representation of if we give it as post-exposure prophylaxis, and C is a representation of what happens when a lot of the antivirals that have been tested with RSV are then triggering on the PCR positivity. So in this case, we take swabs or washes twice daily, and we measure the PCR content of those. And when they're positive for RSV, that triggers them for dosing. So then 12 hours later, they're given the antiviral, and then we can look at what's going on with the disease. This is a very robust way of doing it, but actually it also doesn't translate as well as it might do to field trials, because of course in field trials it's very unlikely you're going to get a volunteer onto the trial that early and be able to get the drug in that quickly. So we're very much advocating at hVIVO to now look at this other later administrations of the antivirals, which is depicted in D in this graph. And the patient perception questionnaire could be just one way of doing that. So if we ask the question and look at not triggering by PCR, but actually triggering by when the subject perceives that they have a cold, which is, of course, what would happen in a field trial, then that is mirrored very nicely in D here. And that's why that patient perception questionnaire can be really valuable in updating these challenge models for antiviral testing to make sure they better represent what can be tested in field trials, and therefore better predictive of whether that product's going to be efficacious or not. And I mentioned right at the beginning that not just have we got a brand new RSV-B model, but we're also really proud that we've got a hMPV model as well. And I've got some sneak preview data for you here. So what we can see here is also very good virus, really good levels of disease. And what we're seeing is good, robust infection rates, over 70% infection rates. And again, this compares very favourably to the RSV-A successful model that we've got. So I'm delighted that we've not just got an RSV-B model, we've also got a brand new hMPV model, also ready for vaccine or antiviral testing. So in summary, we've got a really robust RSV-B model, a robust hMPV model, both excellent safety profiles, ready for testing, and we can move forward with them very nicely. So thank you very much for your attention.